Take a gene is defined as the functional unit of inheritance. As you know that there is no ambiguity that genes are particularly located on the DNA sequence. It is difficult to literally. Now define a gene in terms of DNA sequence. Now the DNA sequence coding for transcription RNA or rRNA molecule also define a gene. So RNA was defined into classified into three types: tRNA, rRNA and mRNA. Okay. However, by defining a cistron, what is a cistron we will learn in the coming class, in the coming point. Cistron as a segment of DNA, imagine it, and which is coding for polypeptide, and the structural gene in the transcription unit could be said as a monocistronic. What is that called? Monocistronic, mostly in eukaryotes. Whereas if you see the polycistronic unit, mostly in bacteria and prokaryotes, polycistronic condition is seen. In eukaryotes, monocistronic structural genes have interrupted coding sequence, and the genes that are located in eukaryotes are splitted because the coding sequence coding sequences and expressed sequences are defined as exons. Exons are nothing but they are said to be those sequences that appear in a mature or processed RNA, and cistrons are appeared in a immatured RNA. Exons are interrupted by introns. Okay. Now introns intervening sequences, which means do not appear in mature process of RNA. Okay. The split gene arrangement further complicates the definition of a gene. Split gene in terms of DNA segment, okay, inheritance of a character is also affected by the promoter, okay, and a regulator sequence of a structural gene. It is also affects the inheritance process of a particular character. Hence, sometimes, if you carefully observe, sometimes a regulatory, and if you see the regulatory sequences are loosely defined as a regulatory genes, okay, which regulates the process. Okay, even though these sequences do not code for any RNA protein, they will not code for any RNA protein. Okay, in the next coming video we will see one more important topic, and stay tuned. Thank you.